because we can't do a lab until we start the next chapter. So this slide right here, this one is pretty much showing you what we've talked about, where we're going to have that messenger RNA strand, the ribosome units come and attach to it. We're going to have tRNAs. As the tRNAs come in, we continue to build our protein. So as we build our protein, one of the things that becomes important is making sure that we get them connected together correctly and that we form that structure correctly. So let's see what happens if we try to play this video.
The ribosome now advances a distance of 1 codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid. Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
And then it just waits for another messenger RNA strand to head out and just start the process all over again. And the stringing together of those amino acids is going to put together our different proteins. Now, this would, of course, be putting together proteins like, for example, hemoglobin, which is found in the blood, okay, insulin, all these things that the cells have to do. So, when we look at how we control the expression of the DNA, when we talk about the all nucleated cells except germ cells are the ones that carry out meiosis. So those are going to be your ovaries and your testes. With the germ cells, it's really interesting because if you think about egg and sperm meet, fertilization occurs, we begin the development of the offspring, and then from that point, everything else is done by mitosis. So it took 2 meiosis cells for the mitosis to start to create the organism that's going to survive. That's pretty cool. Now, when we talk about the development that occurs after egg and sperm meet, well, we're going to find that as development takes place, we're going to have differentiation. Everything that will become something, skin, muscle, nerve, whatever the case might be, it's going to have to be, it's going to be dependent on a chemical signal. So the chemical signals really do make you. who you are. And it forms, you know, male versus the female. It forms whether you're going to have two fingers, you know, that'll stay together, whether all of them are, I mean, it's just amazing. Now, as we continue differentiation and continue forming into the offspring, and as the offspring survives, born, that sort of thing, every cell of your body has a lifetime. Okay? Every single cell has a lifespan. Now, that rate of protein synthesis is going to be dependent on chemical signals. And chemical signals will also affect the lifespan of the cell. Some cells have a very long lifespan, some very short. For example, skin cells. About every 24 hours, because, you know, we're constantly shedding them, so we got to have it continuously move up. Intestinal cells, once again, roughly about every 24 hours, need to renew because they are constantly getting sloughed off as we move materials through the digestive system. But if you think about a cell such as a nerve cell, one that's not going to duplicate, it's going to be stuck in a particular segment of its lifespan, and all it's going to be worried about doing is making its product. That's it. It doesn't care about, that's what its concern is. And when we begin to look at cells of the body, the metabolism, that one of those two systems that I mentioned, we've got the nervous system, we've got the endocrine system. These two systems pretty much run the body. Believe it or not, thyroid hormone. What do you know about it? Ever heard people be like, well, I got a slow, you know, a hypothyroidism or hyperthyroidism, blah, blah, blah. Yes, that can be very true. But why would either of those situations affect other cells of your body? What is thyroid hormone, for example? What does it do? Do you know that it goes to every cell of your body? Muscle cell, nerve cell, intestinal cell, goes to almost every cell of the body because it affects the metabolism of the cell. Basically, the ability of that cell to get and use nutrients. So if you have a thyroid that is hypo, are you getting too much or too little? Too little. What types of symptoms do people have with hypothyroidism?
Weight gain.
Weight gain? Sluggish? Tired? Cold?
Retain fluid.
All right. That is affecting a lot of things in your body. What about hyper? What are some of the symptoms of hyper?
They're real skinny.
Tend to be very skinny.
Hungry all the time.
Hungry all the time. They can eat a lot of food. Their heart rate's usually high. They usually are very hot natured. Okay? But there you go. You have something that's coming all the way from up here, affecting cells in other areas of your body. Pretty amazing. So we have to be able to, basically there's going to be lots of things that affect the life cycle of a cell. Now, when I talk about a life cycle with the life cycle of the cell, that means from the time of the cell getting duplicated, or the cell getting created pretty much, okay, until it goes through cell division. Now, the life cycle includes interphase, mitosis, no. Cytokinesis, even though it's important in the life of a cell, it is not part of division. But what it is division of the cytoplasm. Now here's what happens with cells. Okay, so egg and sperm meet, fertilization occurs, mitosis begins to take place. And then you end up with that little organism that you spit out of your body, okay, that's going to begin to grow into whatever size they grow into, and go and just keep, everything just keeps going, okay? The only time that a cell actually carries out its processes is during my G1 phase. The only time that the cell is doing its job is right here. This is part of what we call interphase. These stages right here are cell division. Do you understand what I just told you? In the entire life cycle of the cell, there is only a very small time frame that it actually does what it's supposed to do. The rest of the time, that cell is in preparation for cell division. Once that cell comes to, okay, and it's in interphase, and we're in G1, everything the cell's supposed to be doing takes place. But then the cell gets to the point where it says, you know, I got to split now. And it goes through making a copy of its DNA. This is not the same thing as making a protein. The synthesis of DNA is making an entire copy of those 46 chromosomes. Okay? Now, if we think about that for a second, I'm going to have 46 chromosomes, 23 pairs. Now, in the cell's lifespan, when it's at 46 or 23 pairs, it's going through G1. But once G1 is done, the first thing that takes place is it is going to duplicate the DNA. It is going to make a copy of every single one of these strands. So making a copy of every single one means that I double the number of chromosomes. So at the end of my S phase and interphase, I now have 92 chromosomes or 46 pairs. Okay? Now, if mitosis did not occur, cells would just keep duplicating. All right.